Hello, my name is Max Saito. I'm a scientist at the Woods Hole Oceanographic Institution, and I'm presenting a talk today called Why Environmental Nutrient Stress Biomarkers Work, Protein Transcript Correlations, and Review of Biomarker Discoveries. I've got a couple of crystal structures of molecules that are relevant to this talk here, a ribonuclease that destroys RNA and an inhibitor that surrounds it, and a protease that destroys proteins. This one's actually from COVID-19, as well as a small inhibitor inside there that will be, their purpose here will become relevant, as you'll see later on. And I want to acknowledge my co-authors on this talk as well. So as someone who works on proteomics, we're frequently asked, do RNA and proteins correlate? The central dogma of biology, of course, is that DNA is used to produce RNA, which is used to produce protein. And there are biomolecules and enzymes behind it that affect the potential correlation. DNA polymerase is used to make DNA, RNA polymerase is used to make RNA, and of course ribosomes are used to make proteins. But the two molecules I showed on the previous slide are involved in the degradation of RNA and in the degradation of protein, respectively. And in biomedical studies, it's noted that correlations between RNA and protein are frequently missing. And here's an example review paper and the relevant text. And their main conclusion is that there's probably other levels of regulation that are at play, which explain why they don't see correlations on global RNA and global protein studies. And those being that there are other regulatory processes such as epigenetics and post-translational modifications and other factors. It's also worth pointing out that those studies are working on complex multicellular organisms with multiple tissues, and in particular internal tissues that don't respond to external stimuli, environmental stimuli. And that's very different from those of us in ocean sciences and environmental sciences, where we're often studying single-celled organisms that directly interact with the environment. And so even small colonial organisms, most of the cells are interacting with the environment and then respond to environmental stimuli. So just from the very beginning, there's probably a lot of big differences. And when you look in the literature at studies of marine organisms, marine microbes that compare the transcriptome and proteome, you often see this trend that I'll show here in the next few slides, where in this case we have a proteome in log2 fold change space, so 2 treatments, in this case phosphorus limitation in this study led by Sonia Dyerman et al. That our lab contributed to. And that's compared to the transcriptome, again log2 fold change. And you'll notice that genes associated with phosphorus metabolism and glycolysis and ribosomes tend to show some relationship, some correlation, whereas there's a large cluster of everything else that's basically hovered around the axis. Here's a nice study from the Giovannoni lab looking at nitrogen limitation in SAR11, Pelagibacter, and they see something similar. Some nitrogen responsive genes and proteins and transcripts relative to sort of a cloud in the middle. We also observed this. This is a paper by Sarah Bender from our group on Phaeocystis antarctica under iron limitation. And again, a similar trend. You can see sort of iron-related proteins at the edges, but sort of a cloud in the middle. And then this was done on 2 strains. And on the second strain, it's flatter response, mostly in the protein axis, which is the x-axis in this study. And less variability on the y-axis, the transcript axis, and we think that's because this was actually harvested at higher biomass, and so it may have been in late log, and the transcriptome might have been more muted as a result. So as part of this study, Nathan Walworth analyzed 8 different treatments for 3 different nutrients for trichodesmium protein transcript correlations, and found in each of these 3 nutrient cases that there was a correlation that existed here for iron stress for 3 different iron related genes using a Pearson analysis and R-squareds that are 0.8 or higher. And likewise, phosphorus stress showed some nice examples of correlation as well for a variety of phosphorus related genes. And then even in the photosynthesis light reactions, there was some relationship too. And then these are the CO2 concentrations shown here in iron and phosphorus co-limitation. So across all these treatments, we saw some good correlation. But interestingly, some of the metabolism genes didn't show this correlation. Instead, we're seeing some dynamic range in protein, but less so in transcripts in these cases. for TCA cycle amino acids and IMB biosynthesis across these 8 treatments. Similarly, Kyle Frischkorn and she and Haley and Sonia Dyerman did a trichodesmium transcriptome proteome analysis for phosphorus limitation and saw dynamic changes in the phosphorus related proteins and the ribosomes, which may be related to growth, for example, in some other systems, again, with this cloud in the middle. So how do we explain this? I wrote a single gene RNA protein box model to model what might be happening. And it's a relatively simple model where we have some signal. This is basically meant to be the environmental stimulus, some environmental signal that triggers a sensory system and a response system, regulatory system. For example, a 2-component regulatory system like a histidine kinase system. And then that results in the production of RNA, which then results in the production of protein. Then there are sinks for both RNA and protein. These are, of course, the degradation pathways, and then we can write out this whole system by these 2 equations, where production of RNA is proportional to that signal times a first-order rate constant minus the degradation of RNA, again, a first-order rate constant relative to the RNA. And likewise for protein, we have production of protein is proportional to a first-order rate constant, and the concentration of RNA, subtracting the sink of protein loss for concentration of protein times another first-order rate constant. And here are the parameterizations then for this model. And we don't need to read all of this now, but I mostly want to point out 2 key parameters. And again, it's those 2 molecules at the beginning of my talk. The RNase and the protease parameters turned out to be key to how this models functions. They're difficult to measure and there's not a whole lot of data for them. For the degradation of RNA, we're using the half-life from E. coli that was measured by tiled microarrays that has a half-life of 6.8 minutes. There was a prior DNA protein RNA model developed by Marianne Moran's group, and they used the 1.5 minute half-life that I think comes from the BioNumbers number repository. We recognize that these numbers are probably on the high end. They probably should be lower, and we've been doing some bulk RNase activity measurements trying to get at what a more appropriate number may be. But I think no matter what the number is, the patterns won't change necessarily from what I'm going to show you. And similarly, there's a degradation of protein, and we use a number from the literature for yeast in this case, and that's got a half-life for proteins of 6.9 hours. So it's significantly longer than that of RNA, as you would expect for a functional molecule relative to a signaling or information molecule. And I think implicit in this model and in our conception of the transcriptome and the proteome and all omes in general is that we assume that organisms are getting perfect information and that they have a perfect response. And I think that's a pretty questionable assumption. Perfect information and perfect responses require a lot of resources in sensors and regulatory systems. And I think it's a really fun question to start wondering how life is wired to produce omic patterns that we observe. And that really gets at the underlying systems biology of environmental omics. And former grad student Noel Held has a nice paper on beginning to discuss this, and in particular, the 2-component regulatory systems. So how does our model work then? And how do the results look? And so there's basically 2 scenarios that are shown. One with a 1% basal signal shown here, and then one with a 20% basal signal, and you can see this starts at 0.2. And then on top of that, we have basically 3 daily signals shown here. Each day is 1440 minutes, the x-axis is minutes. And then these basal signals and environmental signal cues, the 3 environmental signal cues, then result in the production of transcripts, and that's pretty much instantaneous. And then their degradation is quite fast because of the fast short half-life of those molecules. And then because of the basal signal level, we have more transcripts persisting. And I should point out that these are transcripts per 100 cells. Since we're modeling a single gene, it's easier to use a number that's 100 cells so that there's some decent numbers here. And then likewise for proteins, so those transcripts then result in the production of proteins. And there's a little bit of a lag between the production of RNA and the production of protein as defined in the model, and a production rate that's defined in the model as well. And then once the signal disappears, there is some decay that's set up by that half-life of protein in the system. If you have a basal signal, then you begin to accumulate, well, in both cases, you begin to accumulate protein, but with the basal signal, you accumulate more and you sort of have this steady state with some bumps on top of it that come from the environmental cues. And so here you can really see that the faster RNA degradation creates a very different profile of transcripts than proteins, especially if you have a basal signal versus the basically insignificant basal signal, 1% basal signal. So that's what the inventory of transcripts and proteins looks like. But as I showed you in all those correlation plots, all the results typically are presented in fold change space. And this creates some biases that I think I want to point out here. And you can see that, so basically we have our fold change when we have our low basal signal, and then the environmental stimuli coming along, and then you have a 20-fold change in transcripts, a huge change in transcript abundance as a result. Even when you have that basal signal, you still have a roughly 2-fold change in basal signal based on the signal input we provided in this model. In protein fold change states now, you can have a large dynamic range when you have no basal signal, 15-fold change relative to time 0. But once you allow that 20% basal signal, as I mentioned before, you accumulate, you start increasing and you have a steady state protein inventory. And so the fold change then becomes smaller. So our fold chain is, 2-fold relative to time 0. But if you took it relative to day 1 instead of day 0, you're basically going to get a 1-fold change, right? Or no change, because you're comparing it to what's close to the steady state, or plus these small environmental stimuli bumps here. And so the fold change space is biased towards detecting RNA because of that rapid RNA decay and the long protein decay. Here is a video showing the model and what this now looks like. So just to orient you, the video is looping, so don't worry about catching it. We have transcripts going here, and for solid line is 1% basal expression and 20% basal expression in the dashed line. So you can see those are the patterns we talked about before. And over here is the protein dynamics for the same system. These are just the inventories of transcript and proteins per 100 cells. And then down here now are the same results now plotted on log2 fold change. And you can see that basically RNA production is instantaneous at the very beginning, and then proteins catch up. So that's this little bit down here, don't get confused by that. And then as the environmental stimuli comes, you get a big jump in RNA, and then that begins to translate into protein production. As the protein is made, then the RNA is degraded, and then the proteins degrade, and so you start coming down this axis. And then on day 2, you start heading out again, right? And so you can see that if you observe a correlation, it's because you're sampling on this 1-to-1 line when there are both transcripts and proteins present. When you switch to the 20% basal model, you can see the same pattern holds true, but the dynamic range is much smaller, right? It's really much more compressed around the origin because we're basically measuring the ratios relative to an accumulated inventory. And so you're really clustering around the origin, right? And so your fold change relative to day 1 is when you have a 1% basal, you can get a really large fold change in both protein and RNA space. And if you think about environmental parameters, that's what we often have, right? We often have something that has a very low basal expression, and then it's phosphorus stressed, and then it kicks out a large amount of alkaline phosphatase or something, and then you get a large fold change in both RNA and protein. Even if you switch to day 2, that's because of the decay and the low inventory that holds true largely for a large fold change. But once you have a large basal range, that becomes much smaller, right? Because you basically have an accumulated inventory, and that's more the case for metabolic proteins. And likewise, then if you start normalizing to the second day, or when you've really started to accumulate stuff, even that day 2 drops down as well. So basically, briefly on this one slide here, I want to show how there are a lot of nutrient responsive proteins that serve as biomarkers for nutrient stress in marine algae and bacteria, for iron and nitrogen and phosphorus, vitamin B12. We have one for zinc and cobalt that Risk Kellogg in our labs is about to publish, and other vitamins and other things yet to be discovered, right? And we want to point out that these environmental biomarkers appear to work really well for large-scale surveys, but there's more biomarker discovery and characterization needed. We've helped find the B12 and the zinc cobalt ones in recent years, but there's probably other ones that are out there that could really help to characterize systems, or maybe ones that don't exist as well. And I want to point out there's other talks by lab members such as Risk Kellogg on zinc cobalt diatoms at this meeting, and Anneliese Meyer on N and P biomarkers in the North Atlantic. And here's just an example of some sections. So to conclude, environmental biomarkers work for genes, transcripts, and proteins that respond well to environmental stimuli. And RNA protein correlations work for biomarkers, but not for all genes. And why is that? And we developed a model, RNA protein model, that's a MATLAB, and we'll make that available on GitHub shortly. There's a correlation that occurs when timing overlaps for the protein and RNA inventory. And it's observed well for biomarkers because they have a low basal expression. They're basically not abundant when there's not scarcity of a nutrient, and they become very abundant after the scarcity hits. But less so for metabolic proteins because they have a large inventory, right? And so your dynamic range is much smaller. And that's really attributed to our over-reliance on fold change metrics, where that can hide correlations and large changes in copy number when the inventory is large. So you may have a 10% change in copy number, but it still may be a huge signal in terms of cellular resources. And the way to solve this, of course, is to switching to absolute quantities. So thank you very much for your attention.